Hello, everyone.
Yes, we can hear you, Dr. Oladipo.
Okay, good afternoon, everyone. It's nice to be on this platform this time of the day. And I'm very glad that we are collaborating with OmicsLogic, the platform, Biotech, and for this upcoming course that is starting next week, which is tagged as application of microbiome and metagenomics analysis in life sciences. Next slide, please. Yeah, bioinformatics, what we do at Helix, as you have seen, we train, we support and provide bioinformatics consultancy services to meet the need of researchers. We have bioinformatics analysis and planning. We do data management and analysis. We offer workshops and training on various bioinformatics topics for researchers and institutions. And that is one of the things that is ongoing now because we are collaborating with a known and reputable organization that has been involved in bioinformatics training. And we provide resources and allied services for various research projects. Now, I'll be talking on metagenomics. Metagenomics is a study of metagenome, genetics materials recovered directly from environmental samples, such as soil, water, or physics, and some other things. While microbiome is a study of metagenome, genetic material recovered directly from humans or animals. Then how can we apply this metagenomics? To let you know that this training we're about starting is very, very essential for you. For anyone, it can be applied in agriculture, in energy, food and nutrition, biotechnology, biosensor, environment, other industries, medical miscellaneous. I have one of the work I lay my hand on as regards to agriculture that I discovered that a metagenomics analysis of a soil sample can guide us on the appropriate crop to be planted on it. And this is an essential training that I employ all of us to take part in it because it is very, very important. Thank you once again. Viewers, I think you can take up the lead again.
Thank you so much, Dr. Kolawole. And Aditayo, for your kind words and for organizing this workshop along with us. I would now like to say a few words about our OmicsLogic team and our collaborators who have made this training possible. My name is Beepsha Biswas, and I am the OmicsLogic Community Manager responsible for daily interaction with thousands of users like you who are part of our bioinformatics community managed by Pine Biotech. Over the course of the past several years, our team has been collaborating with a number of academic institutions all around the world that have participated in content evaluation, curriculum review, and project design. As a result, the OmicsLogic programs have been thoroughly evaluated and are known to lead to excellent outcomes. The goal of the OmicsLogic programs is to provide training in bioinformatics, enabling independent research guided by mentors and peer example, developing a growing community with shared passion for data-driven research and appreciation of citizen science, enabling students, clinicians, and faculty of all backgrounds to develop novel and independent research using latest technology in life and data sciences. OmicsLogic Metagenomic Training Program will be guided by trainers who have ample experience with bioinformatics in both academic and industry applications. I will now invite Ilya Brodsky, our CEO of Pine Biotech, to speak about metagenomic sequencing and the upcoming program. Over to you, Ilya.
Thank you very much, Beepsa, and welcome everybody to this webinar. I'm excited to participate in this webinar because I think that the microbiome provides somebody
poor network.
Thank you very much. So please keep your microphones muted so that we can all participate here. So metagenomics, I think, is an exciting opportunity because it really provides a snapshot into how these communities that live and function together as almost like an organ could be studied using next generation sequencing. So we can gain insight into composition, species diversity and richness, and understand the functional applications of metagenomics. So in the context of this program, we're going to find different examples where we will see the application of microbiome. We will learn how to analyze metagenomic data, gain insight from data by mapping it onto reference databases, and we will guide you on how to select a project of your own that could relate to either clinical, psychological, maybe soil and environmental samples. So we'll provide a number of curated data examples for you to choose from to work on a project of your own. So let's talk a little bit about the history of metagenomics. Why is metagenomic sequencing such an impactful technology? So bacteria, or the microorganisms in general, have been discovered back in the 1800s. In fact, when bacteria was discovered to cause different pathogenic functions in clinical diagnostics, the first way to study bacteria was to culture it in the lab. And so with the invention of the microscope, when the microorganisms have been detected, then through culturing, we understand how bacteria, different strains of bacteria could have a major impact. But today, with the advent of next generation sequencing, we can study these microorganisms without isolating them in the lab and culturing them. We can actually study the full composition of the communities that they live in. And a big part of understanding those communities relates to the study of the microbiome using computational technology. So when we say metagenomics, we're really talking about analysis of metagenomic data using computational tools. Now, as many of you probably know from personal experience, studying the microbial communities and trying to address how those communities colonize different parts of our bodies is really something that requires an understanding of how they function and grow together. So here we have an example of skin, and in the pores of the skin, you can see how this bacteria can colonize, forming a film, which is very difficult to wash off and disrupt. And that is one of the properties of the microbiome, that they form this relationship with the host and learn how to form a film that is difficult to wash off and difficult to remove because it is integrated into the epithelium. And so when you think of that property of the microbiome and you start thinking about different surfaces and organs that this film can develop on, we now have over 80,000 different publications about the microbiome function in different places of the body. So here you can see a few examples of how diverse these communities can be, but typically a distinct microbiota, a community of the microbes, is formed by dominant bacteria, that you can see here a few examples of those dominant bacteria, and composition, which means that proportions are stable. And those proportions could be disrupted by different conditions and challenged by different environmental challenges. And we'll talk about that in a few moments. But what's interesting is that in those 80,000 publications or more that have been published on the role of microbiome, more than half of those relate to the gut microbiome. Now, the gut microbiome, as is fairly obvious, is the densest community of microbiota in our bodies. And that is also the easiest community to sample because we can take fecal samples. So we can observe fecal microbiota throughout the lifetime of a person. It's easy to sample, it's easy to analyze because we have such clear access to that. At the same time, there have been some recent studies that have shown that that is not the only microbiome that plays a huge role in a variety of different conditions. So let me go through some examples. As you can see here on the screen, this is a timeline of major microbiome sequencing initiatives that start around just after the sequencing of the human genome. And so one of those is obviously the gut microbiome, but then additional ones include the skin, the lung, and even the brain microbiome. So the lung microbiome. The microbiome of the lung has less bacterial biomass, so it has less proportion of bacteria as compared to the gut, but it displays a considerable diversity. The composition of the lung microbiome is determined by elimination, immigration, and relative growth within its community. So when you think of the microbiome of the lung, imagine that certain bacteria are easier to come in, leave the lung area. And so we can see the difference, for example, in chronic conditions. So many forms of chronic lung disease demonstrate exacerbations that drive disease progression and are now poorly understood, but there's growing evidence that supports ways in which microbiota dysbiosis, or the disruption in the composition, can influence the host defense and immunity, and in turn may contribute to disease exacerbations. So it is key to understanding the pathogenesis of chronic lung disease that may reside in deciphering the complex interactions between the host, the pathogen, and resident microbiota during stable disease and exacerbations. So lung is a major microbiota that continues to be studied extensively. The brain is a novel area of microbiome studies. Over the past decade, the development of these sequencing technologies have enabled extensive characterization of microorganisms, including in the gut, lung, skin, and now in the brain. And so because we know that the microorganisms that we coexist with can influence our physiology, immune system, even behavior, we know that the brain could be affected by dysbiosis or changes in the microbiome. But previously, it has been thought that the brain is a sterile environment protected from bacteria and other pathogens. And so now there's first evidence that has shown that, in fact, microbiome of the brain could be affected. So I want to point you to an article. There is actually a discussion in the literature around the role of the brain microbiome. And here you can see some evidence of sequencing reads mapped to individual bacterial presence. So this is very interesting because, as I mentioned, the brain was considered to be a sterile environment. So not only that, we also know that there are other types of microbiota changes related to different diseases. And I think this is a very exciting field. And as we can see here, there is a growing number of publications continually expanding to offer us insights into a variety of different types of microbiomes. So you can see that about 20% of US investment in research has gone to 2 phases of the Human Microbiome Project, which is creating the research resources needed to study the human microbiome in different areas of the body and the different organs. And the goal of this Human Microbiome Project, launched in 2007, is to create a toolbox of reference data sets, computational techniques, analytical methods, and clinical protocols. So we can see how the investment in developing novel technologies and also an understanding of what microbiome is and how it plays a role in human disease has also enabled us to ask very specific questions about the role of the microbiome, which actually play a major role even in the current pandemic. So here you can see an example of recent studies about the role of microbiome in the infection. And it is hypothesized that the changes in the microbiome have an association with more severe disease progression of COVID-19. So it's a very interesting correlation. At the same time, this pandemic intersects with a decade-long decline in microbial diversity and ancestral microbes due to hygiene, antibiotics, and urban living because of the hygiene hypothesis. So what has happened over the past several decades, more people live in cities, there's more uniform level of sterilization, access to sterilized food, access to antibiotics and different treatments that reduce the overall diversity of the microbiome. As a result, the hypothesis is that our body is less exposed to the diversity that could be very important to train our immune system to recognize different pathogenic microbes. So the current pandemic control measures and practices are hypothesized to have an even broader and potentially long-term effects on the human microbiome, right? So there's so much sterilization of hands, there's so much protection from the air that we breathe and the different public situations that we visit. So how will this affect the long-term immunity is a question that needs to be studied. And so much is still uncertain or unknown about the virus and how it's going to affect humans in the future, but we understand that we need to start investing now in our understanding of the role of the microbiome and how it will go through with this pandemic. So to summarize, the function of the microbiome is to help with the differentiation and the development of the immune system, to assist with food digestion and nutrition, to perform energy generation and conservation, help with metabolic regulation of fat storage, process environmental chemicals, maintain the skin and the mucosal barrier, and the prevention of invasion by pathogens. So these different functions in terms of the human health are critical when we study the microbiome. How can the microbiome be studied? It could be studied with two primary technologies that are available today, sequencing and mass spectrometry. So when we speak about sequencing, we can look at particular pathogens that are either isolated or by sequencing everything that is in the microbiome, mapping it onto the reference DNA, or we can also analyze metatranscriptome when we think about what products are going to be produced by these microbes. When we speak about mass spectrometry, we're really talking about detection of particular metabolites or proteins that could be analyzed by their mass composition. So we can look at these spectra and design assays to study what products are and what signaling pathways occur in these diverse and complex communities. So one of the technologies that we will focus on in this program is metagenomics, where we will look at the composition. As I mentioned before, the difference between different other types of omics technologies and metagenomics specifically is that while other types of omics technologies are focused on studying a single organism, a system, here we are going to study how communities of microorganisms form a distinct functional community, and what does the dysbiosis or the change in that composition tell us about the condition that it produces. And so here you can see an example of two ways of studying the microbiome, either looking at operational taxonomic units that we can characterize the composition, or culturing and still use sequencing to understand what amount of different areas in the on this genome are being produced. So we'll talk about these different ways of studying metagenomic data, and we will also learn about how to prioritize different hypervariable regions in the metagenomic sequencing data so that we can accurately detect how different communities that have not all been annotated could still be studied for composition. So we'll talk about the taxonomic classification of bacteria using the 16S ribosomal RNA and the understanding of taxonomy as far as composition in different organs of the body and comparison between different organs in different conditions. So ultimately, our goal with the analysis of sequencing data is to be able to apply the sequencing data to understand the host response, to study the microbiome that includes the bacteria and the viruses that are present in the bacteria, and associate those with clinical or phenotypic data. And so that will be the ultimate goal of this program. And we will see how similar analytical thinking and logic applies to the study of omics data and integration of that omics data for clinical and therapeutic applications. So that would be one of the primary objectives for this program. Let me briefly mention a few project examples that we're going to be using. We have prepared a number of curated data sets, publications that have been digested and turned into tutorials, and we will discuss these at different stages of the program. So one of those, I mentioned already, the Human Microbiome Project. One of those is a part of that Human Microbiome Project is called the American Gut Project. It's a citizen science project where we will learn how to take that data and generate a report, just like what you see right here. This is a report that is produced by the American Gut Project participants, and where you can see the diversity of different sample composition and comparison to other types of samples that are similar to yours. And you can also compare those to multiple tissues in the body, ages and populations, and other factors. We will also talk about the gut-brain axis, or how gut microbiota can affect our levels of anxiety and behavior. And we will talk about methods of elucidating specifically the role of the microbiome by focusing on animal models. So in animal models, what is useful is that we can study how distinctly a portion of this complex system, so just the microbiota, affects different types of behavior. So you can see here that even though we can get insights from patient data, in animal studies, we can design mechanistic studies that will really illustrate what is the role of the microbiome and how that applies to a variety of interventions. Interventions using diet, interventions using different types of antibiotics, intervention using exercise and other factors. And it's important because anxiety is just an insight into overall psychological conditions and even clinical conditions that arise potentially as a result and having an impact by the microbiome. So here's an example of China approving the first Alzheimer's treatment that targets the microbiome. And you've probably heard of many other examples of interventions using fecal transplantation and other types of interventions. So here we see a number of cancer therapies that have proposed to be affected by the microbiome composition. And therefore, we understand how the metabolic potential of these microbes and the products that they produce is an important area of study for us to have more personalized and more effective treatments for different types of patients. Now, as was mentioned already before me, it's not only that the microbiome is affecting human health and disease, we also will talk about microbiome in agriculture. So how can we actually study the composition of soil and different bacteria that live on the roots of plants? So there's the soil microbiome, the bacteria and the microorganisms that live in the soil. And then there's also specific communities that form around the root nodes. And those play a major role in the way that the plants can absorb different nutrients and interact with the environment. And so we'll look at examples where the interaction and the composition of these microbial communities can play a role on the production capacity, so how much fruit is being in plants, how well they grow, how resistant and resilient they are in different harsh conditions, and also, as was mentioned, how to select the best soil for different crops and for different objectives. So I hope that this will be an interesting program for all of you to consider joining. And to speak about how to join this program, I want to pass it on to Dr. Mohit Mazumdar, who will guide you through the registration process.
Thank you, Ilya. Yeah, so I'll just share my screen. Okay, hello everyone. My name is Mohit Mazumdar. I'm one of the global representatives of Pine Biotech. And our mission here is to simplify complex bioinformatics research and reach out to the student and research community to participate in advanced data-driven biological research through our modular and intuitive multi-omics T-Bioinfo platform and our coding and educational portals. So this program, OmicsLogic Metagenomics, it is a specialized program that not only takes you through the live sessions, courses, example publications, and datasets, but also gives you an unique opportunity to apply this knowledge to a real metagenomics project. So as in all of our OmicsLogic training programs, we will be leveraging online sessions such as these, but more towards hands-on activity. And then we will be also using self-guided materials, and that are carefully and that have been validatedly designed and taken by thousands of students across different groups. And then we have practical assignments and tutorials that you can take to learn and go deeper into these methodologies and techniques. So this entire experience is OmicsLogic is in the OmicsLogic training program. Sorry. Okay. So this online program is equally rewarding for students and researchers, and especially suited for project-based learning on a flexible schedule. So all of these sessions that we will be having for this program will be recorded and will be emailed to our participants. Other than that, the program will cover main aspects of metagenomic sequencing, analysis of 16S RNA, and other aspects of microbiology research using bioinformatics. So in these sessions, we will do hands-on experience using the T-Bioinfo platform, and also we will practice through our OmicsLogic platform. And then we will learn about how 16S metagenomic data is processed, and that results in a table of annotated 16S rRNA genes. And then you can read those map counts, and there's that table that could be annotated by taxonomical levels like kingdom, family, genus, species. So learning about all those through hands-on experience that we will be doing through all these sessions, along with the materials that will be shared with you as a form of courses, as form of example projects, or example publications. So at the end of this program, participants will be conducting an independent, there will be an opportunity to conduct with an independent or a group project, where you will be able to work on publicly available data sets and learn from the and learn about the impact of this publications and the research on the microbiome. So the sessions will be around the same time, 5 p.m. To 6:30 p.m. West African time. So our metagenomics bioinformatics research training, and that what we are discussing today, and all of you have joined to learn about, is a research training which is well structured, and it has been recognized by experts from academia and industry as well. So talking more about like how this program would be and what these resources are that I'm talking about. So this specialized training involves a lot of project examples, because learning by doing is one of the best ways to learn. So starting from a level 0 or designed for beginners, so introduction to metagenomics, you'll start from a basic level where you learn about the keywords and the application, how this field has evolved, to a advanced level, where you will be further supplemented by publication derived project examples that are available as educational projects. So the educational projects are tutorials that explains a peer reviewed published research project in a simplified manner and explains the data set to next work on the analysis using the T-Bioinfo platform. So that will be the part of this program, and we will be doing that by also showing you how to do that. So these are already described in the courses and in the project examples, and then we will have the live sessions to supplement that and to answer your questions or the problems that you have faced or any of those issues that would be addressed. So one of the biggest advantage of this program is just that along with the Zoom sessions, you get a very important aspect of mentor support. So our OmicsLogic programs are supported by experts, and they have a rich experience and rich background in both industrial and academic bioinformatics research projects, and both in education and training. So I think most of you have joined today's session, have an interest in metagenomic research. So I just wanted to talk a little bit about metagenomic research, and I know Ilya did talk about the metagenomic research in length. So this research field, it's a newer field of biological research, and it has already impacted us. So I just wanted to show you some examples. So that recent study about the gut microbiome and COVID-19 that Ilya said talked about. So the one of such study is like where the author concluded that the individual gut microbiome may affect the body response to SARS-CoV-2 infection, which means like the gut bacteria might influence both short and long-term effect of the infection, and how the gut microbiome can affect brain health in the case of neurodegenerative diseases that might lead to better and easier treatment for brain diseases. Another example is this segmented filamentous bacteria in gut. So in green, you can see they are overstimulated the immune system of a pregnant mice that have an infection. So that altered its fetus brain development. So that's one of the research aspect. So this field of metagenomics is gaining insights from uh, micro microbes and microbiota, has generated like enormous interest, and not just for academia or academics, but also in industry. So in February 2019, Axial Therapeutics in Massachusetts, they raised $25 million in financing to develop therapeutics for neurodegenerative diseases and neuropsychiatric diseases. Another company is this Finch Therapeutics in Somerville. So they are developing an oral microbiome drug for autism spectrum disorder, ASD. So they announced that was announced in last year September, and they have raised $90 million for that. So there's a lot of research that is ongoing, and there is a lot of requirement in industry and for a postdoctoral level as well for this kind of research. So one of the other aspect is to develop an understanding of metagenomics research experience. So to do that, one needs to have a good understanding of the way data is prepared. So in the context of the program, we will be discussing data sets that are available for analysis and study of the role microbiome plays in agriculture, biomedical research, epidemiological studies of infectious diseases, such as those. So during the last program, and we had this program in September, there were a lot of interesting studies that happened during that program. So that resulted in numerous independent projects. Some of them are highlighted here. So these projects were then peer reviewed by experts. So that's how it works. So towards the end, when you know that how this works and you have an idea and you want to work on a project, we ask you to submit a proposal. So there are already ongoing projects that you can join. There are already projects on the educational side that you can further work on. And the third aspect is the independent project. So many of these researchers who are working on their own research projects, or they are working in their PhD in their labs, they have some research questions that could be addressed using bioinformatics, and that could actually enhance their publication quality and the quality of research, or answer their questions with a complete understanding. So for that purpose, many of these researchers submitted their research proposals, and we went through a peer-reviewed process by sharing those research proposals to experts, and then we got that feedback from them, and then we kind of analyzed it ourselves, and then we have a session with the participants discussing their research proposals. So some of the research proposals were really, really exciting and interesting. Some of them were about the International Space Station and the data that have been collected in the human missions for other long-term missions. Some of them were about finding relationships between SARS-CoV-2 and how metagenomics can help in understanding the virus toxicology and transmissibility. So those are some of the aspects of this program. And without taking much of your time, I would just wanted to say that I'm really excited to be a part of this collaborative research program, and I'm looking forward to meeting the participant to guide through the resources, and then developing logical and conceptual understanding of the bioinformatics research. And to talk more about the program resources and registration, let me invite Beepsha to guide us through the process. Thank you.
Thank you so much, Dr. Mohit, for such an insightful presentation. For different parts of this program, we will leverage Omics Logic resources that you can use to learn more about metagenomics. These will include asynchronous coursework with theoretical material, quizzes and practical assignments, our cloud-based analytical server, where you can analyze large metagenomic data and visualize results, the Omics Logic code playground, where you can practice coding in R and Python, as well as a repository of project example. So let us start by signing up to our platform, which is the Edu T-Bioinfo platform. If you already have an account, you can log into the platform. If you do not have an account, then you can either sign in using your email ID, or you can utilize any of these social media logins to sign into the platform. This will get you started on your own bioinformatics journey. The first course that we recommend to you is the Introduction to Metagenomics, which will familiarize you with the concepts of metagenomic data analysis. And you can also find the link in the chat box. Thank you, Aditayo, for sharing the link. You can also see some of these reviews by previous participants who have taken this course. So this is a free course. Please feel free to create an account and start taking this course. You will also discover that this comes in a set of 3 courses, starting with Introduction to Metagenomics, Metagenomics 1, and Metagenomics 2. And if you are registered for the program, you will have access to all 3 of them, as well as a number of curated data sets prepared as projects, like the American gut microbiome, gut-brain axis, and the role of microbes for skin conditions like acne, eczema, and more, which already Ilya and Dr. Mohit has spoken about. Let us now talk a little bit more about the practical analysis approaches we will cover in the program, like KIND2, DADA2 pipelines, and PhyloSeq. Throughout this course, you will be given directions to access the T-Bioinfo platform for bioinformatics processing and analysis of metagenomic data. The platform includes demo pipelines as well as data management and analysis cloud infrastructure to run bioinformatics pipelines. Different stages of analysis are performed in different sections of this multi-omics pipeline. The visual interface of the TBioInfo platform will enable users to quickly build logical graphs of their analysis and understand each steps significant in running this pipeline. For those interested in implementing the pipeline on their own, kindly keep your microphones muted. Thank you. So for those interested in implementing these pipelines for your own, we will also offer guidance and references that can be used to effectively install all the needed packages for these pipelines. So behind many of these methods are scripts written in popular coding languages such as R and Python. These are explained in tutorials you will have access to on our OmicsLogic platform. These include beginner tutorials on data importing, preparation for analysis and standard visualization of processed data. More specialized packages with publication-ready outputs will also be covered in more advanced lessons. That is why this program is built to provide students of all backgrounds with a clear perspective on big data analytics, machine learning, and artificial intelligence in application to omics data and research, clinical, and industry applications. Our OmicsLogic step-by-step process is enabled by access to complex tools via a simple and intuitive interface, at the same time exposing participants to analysis logic in the context of real data sets and extrapolation of this logic to a variety of projects and data sets with assistance from mentors and technical support. Now let us take a look at some participants from our recent bioinformatics training programs in Nigeria who have been very successful. Here you can see Farida Bashar, who has gained over 10,000 points and have completed multiple courses denoted by the course certificates. So the free course that we have shared also comes with a certificate of its own. So if you are looking forward to earn a certificate, you can complete Introduction to Metagenomics and you will be earning a certificate. Like Farida, Abiosogun Mujib is also working towards completing the courses. And as you can see in both these cases, the participants have a complete profile. So once you're logged into EduTBioInfo, the first step is to update your profile. And an updated profile comes with a full picture and your full name, as this is the name which will be reflected in the asynchronous course certificate that you would be getting. And you can also learn more about these participants via the Edu link. So you can also be one of those participants who are featured in one of our webinars in our social media channels. So please feel free to make sure that your profile is updated. And Aditayo has also shared all of the profiles of these two participants. Your achievements in the program are denoted by the points, as we saw in the last two slides. So once you complete this training, you will receive either a certificate of excellence or a certificate of completion, depending on the number of points you achieve. And this is an effective tracking mechanism. Why? Because the points you receive are not only from the coursework that you do, but also by running pipelines on the TBioInfo server or by using the OmicsLogic platform for running the codes. So all of these platforms are linked together. So all of your activity is tracked and they add to your points. So you, or as well as your faculty, or even if you're coming as part of a university, can track your progress. Even our mentor team can track your progress. To start with the program, please register on the EduTBioInfo portal. And once you are signed in, we will be sending you a link that will ask you to register for the program. And you can visit that link to complete your registration. Now, The cost of this program is $150 USD, and we understand that this may not be an affordable option for a lot of participants here. So we also have a scholarship option for students. You would need to complete the necessary requirements to be awarded the scholarship. Please start by filling out the form that you will see in the chat box if you're interested in applying for this 1-month training program. And now I'm going to be opening this session for questions. And also I would like to ask you all to see a poll right now on your screen where we would want to know more about you. So the first question here is, what is your educational background? The second question is, what are your areas of research interest? So we do know that a lot of you are interested in metagenomics research, but you may also be interested in either of these research areas. So please feel free to share. And the third question is, where are you from? So I will keep the poll on, and we would try to address any questions that you may have. So I see that a lot of the questions that came in are regarding the different resources, or if you can get started if you are a beginner. Yes, you can definitely get started with this program even if you are a beginner, because we will start with basic courses like Introduction to Metagenomics, which will give you an overview of this full metagenomic training and metagenomic data analysis. Can you see the poll on the screen?
Yes, we can see the poll.
Jay Kishan, are you not able to see the poll on the screen? Okay, so I'll keep it on for a few more minutes. And if you have any other questions, then you can please put them down in the chat box. And again, this session is open for everyone to participate in. So please go ahead and complete your registration. We do not have a long time before February 16th, so we would be starting it next week. So thank you again for making time to join today's session. Yes, so
before we leave, Bipsa, I would like to just, there was a question on how to get access to the platform. So maybe I could just explain how that would work.
Sure, Ilya. So should I stop sharing my screen? Okay.
Yes, please. So the server platform offers a cloud-based account where you can store large data sets. You can import data sets directly from NCBI, upload your own FASTQ files and reuse them multiple times, and also analyze, process, and interpret the results. So first of all, when you get to this platform, you will see this link right here in the menu that says Access. So what you need to do is fill out your name. That information comes to us by email, and we will review your application and send you the details. And there are 3 different options to use this. The first one is the free option. So using the free option, you understand how different sections of this platform operate and what types of outputs and inputs they utilize. For example, since we're talking about metagenomics, here we have a demo pipeline for one of the data sets that we will discuss in detail during the program. It is a data to processing pipeline with interpretation using Phyloseq, which is a package in R. So you start the pipeline by clicking on Start. Here you have a little explanation of what the overall idea behind this pipeline is. Then the data to pipeline consists of a number of different steps that are specifically designed to eliminate the noise in the data and make sure that the result, the output, is cleared from all of the contamination, because you're talking about many different unannotated reads from a whole variety of different microbes. Finally, when the data is analyzed based on a database called the Silva database, now you can study what is the relationship between different groups of reads based on their taxonomic association using a phylogenetic analysis called APE, which produces biome files that could be visualized and further analyzed for their composition using this package called phyloseq. So finally, when your data is processed and analyzed, you will see some pipeline outputs, and you can really see what to expect. So what are these OTUs? What is the taxonomy table? What are the different plots that you can get from phyloseq, etc. So this is an example for the free tier. Anyone can register and get this demo pipeline for free. You can see a variety of different examples, including the microbiome, transcriptome, genomic data analysis, mass spec, structural biology, data integration and modeling, data mining and machine learning, and other specific processing pipelines for virology, such as different strains in COVID-19, detection of mutations, and things like that. So this is a full-fledged bioinformatics analysis platform that will allow you to process, analyze, and visualize your omics data. Now, included with this, as I already mentioned, are some packages in R and Python that you could learn how to read, modify, and adapt to whatever you want to do with them, also learning how to code along the way. And so the second resource that Beepsa demonstrated was the Code OmicsLogic Playground. And here we also have a section dedicated to metagenomics. So this is going to be covered again extensively in the program itself, but you will learn how to perform analysis of sequences, visualization, composition, statistical analysis, and a number of other things that are critical for anyone that wants to learn how to make the most out of their microbiome data. So the unique part of this is that you actually get to see the code and try to run it yourself. So you learn about the code, you see the different outputs, and then you can actually try it by yourself. So here we have created a way for us to take the scripts and run them in RStudio on the cloud, and really eliminates a lot of the challenges that people have with setting up their environment. So here, for example, you can see I've analyzed the data set, and I can see the composition of my sample based on proportion or based on abundance. So this is a little bit of an overview of some of these tools and how to register. Again, many of these resources are completely free. So if you don't want to join the program, you don't have maybe the time, or you want to do this on your own pace, you can register here for free. Start learning, and when the time comes, you will see different types of additional in-depth resources, like actual research capabilities of the platform, where you can upload your own data and manage it using FTP. And on the coding, we also have some advanced coursework like machine learning and data science that could get you farther along with the data that you have.
Thank you, Ilya. So there is a question from Ayodinde. Is it possible for one to jump directly on the core metagenomics where we can use the actual R scripts?
Yeah, so as I showed, the same R scripts are offered there. In any kind of processing of data, it's not the scripts that typically limit you. It's the infrastructure. So if the data is big, and many of the current studies include hundreds, if not thousands of samples, and the analysis, you can do them on your computer, but unless you have a server stack, you're going to be waiting for a long time for those pipelines to finish. So that's why we have a lot of these pipelines implemented on our big data, high performance computing infrastructure for you to be able to work with real data sets. But once the data sets are processed and they're ready as a table, you can then do the downstream analysis and visualization in R and in Python. And that is a big part of what we will do in the program as well. First of all, understanding the logic. Why are we doing what we're doing, and what do these visualizations mean? and then doing it ourselves in R and learning how to manipulate the data, wrangle it, visualize, and interpret.
Thank you so much, Ilya. So I would want you all to go ahead and join our social media groups. So we are having an active group in LinkedIn. And if you go through the group, you can see that this is a group not only for students or researchers, but you would see that there are a lot of scientists who are joining us in this group from all around the world. So this gives you an opportunity to be part of a larger community, as well as we have a group in Facebook. So whichever platform is easy for you, please go ahead and join our platforms. And now to clear some of the doubts that a lot of you are having regarding the registration. So if you are already registered on EduT Bioinfo, I'm putting the link in the chat, then this is the link to check out. So you can note that here is a $150 the price. There is a 33% discount that would be emailed to you if you have already shared your email ID or if you were already registered for this program. So you would have to check out via this link, and you can also see the different courses that are included as part of this training program. So please visit this link. And if you are someone who is looking for a scholarship or you require a scholarship, here is a scholarship form. Again, I'm posting it here in the chat box. So please make sure to fill up each of these fields in the scholarship form. And maybe let us take a look at this together so that we know what are the different things that we have to fill up here. So you see, you have email, first name, last name, the company, an organization, your current level of education, graduation year, current CGPA, any other awards. And the reason you're applying for the scholarship, you have to upload your CV. And please make sure that you're also mentioning a reference from your college or university, and also this field. So all of these fields should be filled up for that scholarship to be accepted. And once that scholarship is accepted, or we want to discuss more with you, we would set up a meeting with you and your reference. So please make sure to mention the correct name and email addresses of your references. We'll set up that meeting, and that meeting will decide whether that scholarship is going to be given to you. So please go through this process. And if you're joining us directly, please follow this link. And we will also be sending you this entire link to join the program and send you an email with all the details about the program. And this scholarship may either be a full scholarship or only a partial scholarship. That would be decided while we are having that conversation with you and your reference. So it would be dependent on that meeting that we have with you. And now moving on, I would also want to highlight 2 more programs that we are having very soon. So this program is again a specialized training on precision oncology. So here is the link again if someone is interested in joining this program. This is starting on March 15th. And we also have an ongoing program for researchers and students who are interested in working on their own project. So this is either a 3 months or a 6 months project development program where we work closely with a student to help them work on their own novel project. And our team of mentor guides that student right from the training to the completion of that project. So if you're joining like a 3 months program, we are here to make sure that you can come up with a project of your own. And of course, you would have to give in quite some time of yours to be able to come up with a novel project. While if you are also intending to go beyond just a project and aim towards, say, a poster presentation or even working on a publication, which can be a very long drawn out process. So we definitely recommend 6 months or more to be able to make sure that that project is ready to be presented to everyone else. So this is again another one of the offerings that are available for students, clinicians, or researchers who are interested in attending a research program like this. And with this, I would like to thank you all for attending today's session. And I hope you enjoyed this session and it was informative to you, and hope to see you in the upcoming week in the microbiome and metagenomic data analysis program. Thank you, everyone.